Hello all, welcome to my channel. In today's video, we will be studying about a very important process that occurs in all living organisms, and this process is known as the electron transport chain or the electron transport system. In most of the organisms, there is an additional process that is coupled with this electron transport, and that is known as the oxidative phosphorylation. So today's video will be focusing on this coupled process of electron transport chain and oxidative phosphorylation. Now, when any carbohydrate is broken down, most usually it is glucose, but it could be any other carbohydrate. When it is being broken down, that carbohydrate first undergoes the initial breakdown from a macromolecule into glucose. Then that glucose is converted into pyruvate by the process of glycolysis, which I have already spoken to you earlier. The pyruvate is then converted into or completely broken down into carbon dioxide by the process of the TCA cycle or the Krebs cycle. Now, during this process, during the conversion of glucose into carbon dioxide, there is release of ATP at certain points, that is the substrate level phosphorylation, and there is release of NADH2 and FADH2. These are the three major byproducts that are obtained when glucose is being completely broken down into carbon dioxide. This, what I have just told you, is what we call as the aerobic respiration. So when I say respiration, it is an energy yielding process. The cell is looking for maximum amount of energy by breakdown of glucose. But if we see the ATP that is obtained by substrate level phosphorylation from glucose to carbon dioxide is very less. So the cell needs to produce more ATP, and how it does that is it converts the reducing equivalents, that is the NADH2 and FADH2, into ATP by the process of electron transport chain and oxidative phosphorylation. So how the cell is using the reducing equivalents to produce more ATP is what we are going to look at. Now, in the electron transport chain, there are 5 different kinds of electron carriers. All of these electron carriers are embedded in the membrane. Now, which membrane does it occur in? If it is a prokaryotic cell, like a bacterial cell, then all of this process, that is the electron transport chain and oxidative phosphorylation, will occur on the plasma membrane. Please remember, these electron carriers need to be supported in a membrane. So it is occurring in the plasma membrane because they do not have any organelles. If it is a eukaryotic cell, then electron transport chain and oxidative phosphorylation will occur in the inner membrane of mitochondria. As you would have all earlier heard, mitochondria is called as the powerhouse of the cell. That is where all the ATP is being produced. So in the inner membrane of mitochondria, these electron carriers are embedded, and that is where we are going to see the process of electron transport chain and oxidative phosphorylation. So these 5 types of electron carriers can be roughly grouped as the nicotinamide nucleotides. Now these are the coenzymes which are associated with the dehydrogenase enzymes, and they contain the vitamin niacin. These are being produced in the NADH2, that is the reduced form of NAD, is being produced in TCA cycle, it is being produced in glycolysis, it is being produced in the link reaction. So these are the first type of electron carriers involved, that is the nicotinamide nucleotides. The second type are the flavoproteins. Now flavoproteins are those which contain a prosthetic group of flavin. So they contain riboflavin, and these are also called as flavin coenzymes. Examples of these flavin coenzymes or flavoproteins include FAD and FMN. FAD stands for flavin adenine dinucleotide, and FMN stands for flavin mononucleotide. So both of these contain riboflavin, and they are also being produced. FAD is being produced in the TCA cycle, whereas FMN is present as an electron carrier in the plasma membrane or in the mitochondrial inner membrane. The third type of electron carriers are the iron sulfur proteins, also called as the non-heme iron proteins. The reason they are called as non-heme iron proteins is because here the iron atom is present in pairs connected by an iron sulfur bridge or an FES bridge. So that is why they are called as FES proteins, iron sulfur proteins, or non-heme iron proteins. I just have certain iron atoms which are interconnected with the help of sulfur bridge. So these proteins are also very, very commonly found in the membrane, and an example of this is ferredoxin. Ferredoxin is again an important type of electron carrier that is seen in mitochondrial electron transport or even in nitrogen fixation. In many other electron chains also it is being found. The fourth type of carriers are the quinones. Now, quinones are compounds or moieties which are made up of isoprenoid units. There are different types of quinone based on which cell we are talking about. For example, if it is an animal mitochondria, the quinone is called as CoQ10 because it is having 10 isoprenoid units. So it is called as coenzyme Q10. Or we could even have the ubiquinone, which is found again in the mitochondria. We can have plastaquinone, which is found in the plants. Gram positive and some gram negative bacteria have naphthaquinone. So the type of quinone will vary from one cell to the other. In the further slides, we'll be just showing quinone as Q because the type of quinone is varying from one cell to the other. The last type of the electron carriers and one of the most important ones are the cytochromes. Now there are different types of cytochromes and they were discovered in way back in 1886 by Machman. They have been classified into cytochrome A, B, C, D. So their amount is proportional to the respiratory activity of the tissue. And then there are different subtypes under each one of these, like we have A1, A3 or C1. So the main characteristic of these cytochromes is that they have an iron porphyrin ring as their prosthetic group. So they are proteins, but they are having a porphyrin ring. That is the reason cytochromes are electron carriers with a porphyrin ring, whereas iron sulfur proteins are non-porphyrin ring proteins. They do contain iron, but they do not have a porphyrin ring structure. So these are the 5 types of electron carriers which are arranged in the plasma membrane if it is a prokaryote, or if it is a eukaryote, it is arranged in the mitochondrial inner membrane. Now, how are they arranged? So these electron carriers are arranged in the form of 4 complexes. The 4 complexes are the first complex, which is shown in red over here. The first complex is also called as the NADH reductase. Now, if it is a mitochondrial cell, it is called as NADH CoQ reductase. Like I have already told you, coenzyme Q is the electron or the type of quinone that is found over there. In other cells, it is called as the NADH reductase. The second complex shown in blue over here is the succinate reductase. Again, if it is a mitochondrial cell, it is called as succinate CoQ reductase. So the first one is NADH reductase and the second one is succinate reductase. The third complex, the one which is shown here, complex 3, is also known as cytochrome C reductase. And the fourth one is called as cytochrome C oxidase or oxidoreductase. So cytochrome C reductase is complex 3, whereas cytochrome C oxidase is complex 4. So these are the 4 complexes that are present, and when I say complex, it means there are 2 or 3 electron carriers which are grouped together to form a complex. Apart from these, there are also 2 free electron carriers or mobile elements. The first one is quinone and the second one is cytochrome C. Cytochrome C is present towards the exterior of the membrane. Now, if you see the membrane, this is plasma membrane. This can be considered as plasma membrane as long as we are talking about prokaryotic respiratory chain or electron transport chain. This has to be considered as the inner membrane of mitochondria if we talk about the mitochondrial respiratory chain. So based on what is your requirement, you need to write the names differently. So if it is a mitochondria, this is mitochondrial inner membrane. What is towards the outside is the intermembrane space between the inner membrane and outer membrane. And towards the inside, we have the mitochondrial matrix where the TCA cycle has occurred. If it is a prokaryote, this is the membrane, plasma membrane. Externally, I have periplasmic space and internally, I am having the cytoplasm. So based on your requirement, you can change the denotations or you can write the terminologies differently. Now, the respiratory chain or the electron transport chain is basically a series of oxidation reduction reactions. So in which the reduced coenzymes are being oxidized by oxygen. So the terminal electron acceptor is oxygen, whereas the reduced coenzymes are going to donate their electrons and in the process get oxidized. These coenzymes are donating their electrons to the electron carriers. The electron carriers are carrying the electron. As per their name, they are going to pick up the electron and give it to the next component. Finally, the electrons are being accepted by oxygen. Oxygen accepts this electron and gets converted into water. This is what is happening in aerobic respiration. So along the electron transport chain, the redox potential or the reduction potential of the electron carriers is going to keep increasing, which means the electron donor, or where we start from, NADH or even FAD, they will have a highly negative reduction potential, and oxygen will have a highly positive reduction potential. This difference in reduction potential is what is helping us to move the electrons. So this is having a highly negative reduction potential. It is waiting to donate electrons, whereas this is having a highly positive reduction potential. It is waiting to accept the electrons. And this is what is propelling or helping the electron transport chain to function. So the electrons are going to move automatically or spontaneously from a highly negative reduction potential compound to oxygen, which has a highly positive reduction potential. So let us see how the movement occurs. Initially, the electrons are being donated by NADH2, that is nicotinamide adenine dinucleotide, which is in the reduced form. That is why it is called as NADH or NADH and H+. It is donating its electrons to complex 1, and in the process it gets oxidized back into NAD. Now in the complex 1, that is in the NADH CoQ reductase, there are the electron carriers FMN and FES. So the two electrons which are released by NADH are being taken up into the first complex, or they are being taken up by these electron carriers FMN and FES. First it goes from NADH to FMN, and then from FMN it goes to the iron sulfur protein, that is the FES. From there, the two electrons, so it's always a pair of electrons, the two electrons are passed on to quinone. From complex 1, they move to quinone. Quinone is not bound by any complex, it's a free carrier. Quinone carries those electrons to complex 3. Now complex 3 comprises of cytochrome B, cytochrome C1 and iron sulfur protein. So here we have the two electrons moving from quinone to complex 3, and in the complex 3 we have three different types of electron carriers. From the complex 3, the electrons are being taken up by cytochrome C. Now cytochrome C will transport only one electron at a time. So it will come, pick up one electron from here, drop it at complex 4, again come back, take the second electron and drop it at complex 4. So cytochrome C is going to pass on these electrons one at a time to complex 4. Complex 4, that is the cytochrome C oxidase complex, comprises of cytochrome A, cytochrome A3 and copper ions. Cu2 plus ions are also present over here. From here, this is the terminal oxidase. This is the terminal complex. From here, the electrons are being given to oxygen. Oxygen picks up the electrons that is given by complex 4, takes up two protons from the cytoplasm to form water. This is the flow of electrons from complex 1. From complex 2, the complex 2 takes up electrons from the succinate. So succinate, during the process of the TCA cycle or the Krebs cycle, it forms FAD. Now that FAD is actually a membrane bound complex. It is present in complex 2. So FAD gets converted or gets reduced to FADH2. It donates its electrons to the iron sulfur protein present in complex 2. From there, the electrons flow to quinone, then to complex 3, from there to cytochrome C, from there to complex 4 and finally to oxygen. So the electron flow from NAD is via complex 1, quinone, complex 3, cytochrome C and complex 4. There is no complex 2 coming when the electrons are flowing from NADH. On the other hand, when the electrons are flowing from FAD or from succinate, complex 1 does not come into the picture at all. So complex 1 and 2 are independent of each other. All the others are common electron carriers in both the pathways. That is why they have been shown in purple, whereas complex 1 and 2 have been shown in different colors because they don't mix with each other. Now, these two complexes are going to donate their electrons finally to oxygen to form water. This is the electron transport chain. But I have not told you where the ATP is being produced. And I told you at the beginning that all of this is being done to produce more and more ATP. So let us see where the ATP is being produced, which is described by a process called as the oxidative phosphorylation. Now, this oxidative phosphorylation or the chemiosmotic theory or chemiosmosis was described by Peter Mitchell, who said that as the electrons are moving through the membrane, there is also pumping of protons outside the membrane. So there are 3 points at which, or the 3 complexes at which the protons are being pumped out. Now, because these complexes are driven by electrons, but they are pumping out the protons, hence they are also called as electron driven proton pumps. What do? They are going to pump out certain electrons from the inner part of the cell. In case of a prokaryotic cell, they are pumping it from the cytoplasm into the periplasmic space. Or if it is a eukaryotic cell, the electrons are being pumped from the mitochondrial matrix into the intermembrane space. So the protons are being taken and pushed out of the cell or being pushed out of the membrane at 3 different points or by 3 electron driven proton pumps. These electron driven proton pumps are the complex 1, complex 3 and complex 4. At the other points during the electron transport chain, there is no pumping of electrons. But at these 3 points, there is a bulk of protons being pumped out of the cell. Now at complex 1 and complex 3, we are having 4 protons being pumped out. So when 2 electrons are moving from NADH to the electron carriers or into the complex 1, 4 protons are being picked up from the cytoplasm or from the mitochondrial matrix and pumped out of the cell. The same way, even at complex 3, as the electrons are moving from the complex 3 into the cytochrome C, there are 4 protons being pumped out at this point. At complex 4, 2 protons are being pumped out, whereas 2, like I told you earlier, are being used in the production of water. So at 3 points, we have a total of 10 protons being pumped out, 4, 4 and 2. Now when there are so many protons being pumped out, definitely it is going to lead to an instability in the membrane. So that instability in the membrane or the energized state of the membrane is known as proton motive force. Now, why does that proton motive force occur? When all the protons are being pumped out of the cell, they need to come back in. Otherwise, the cell is not going to remain stable. So when they come back in, they are coming back through the complex 5, or it is also called as the ATP synthase complex. That is what has been shown in green over here. This complex is going to bring back the protons, and when it is bringing back the protons, it is having enough energy to catalyze the conversion of ADP, that is adenosine diphosphate, into ATP, that is adenosine triphosphate. Now this is happening because of an electrochemical gradient. So chemiosmotic theory is explaining how the ATP is being synthesized at the end of the electron transport chain. But how that is occurring is because of an electrochemical gradient. Now, the electrochemical gradient is occurring because when there are a lot of protons being pumped out of the cell, that leads to an imbalance. That is leading to an energized state of the membrane, which is called as the proton motive force. This energized state of the membrane, or it is this force which is helping us to produce ATP. Now, how is that electrochemical gradient being established? As you can see over here, at complexes 1, 3 and 4, I am having protons being pumped out for every pair of electrons. And if you recall, in the entire glycolysis, link reaction and TCA cycle, we have many reducing equivalents formed. So each one of them, when they enter the electron transport chain, at every point, I am having protons being pumped out. This leads to the exterior of the membrane having a high quantity of protons, and the interior of the membrane is going to have an in turn high quantity of the hydroxyl ion. Now, when there is high quantity of protons, one is it is leading to a pH variation. Here, the exterior of the membrane is highly acidic in pH, whereas the interior of the membrane is highly basic in pH. The second thing is the charge variation, electrovariation. External or exterior of the membrane is having a high quantity of positive charge, whereas the interior of the membrane is having a high quantity of negative charge. So this is what we call as an electrochemical gradient. Electrochemical gradient is due to two things. One is the charge difference across the membrane, and the second one is the pH difference across the membrane. Both of these are what are causing the membrane to be in an imbalanced state or in an energized state. So to come back into the balanced state, the membrane is going to bring back the protons through the ATP synthase complex. Now the ATP synthase complex is again a set of proteins, and this is made up of two big subunits. I have a membrane embedded F0 subunit and a circular or a globular F1 subunit, which is projecting into the cell or which is projecting into the interior of the membrane. So this F1 F0 subunit is going to help in transport of the protons back into the cell plus the production of ATP. ATP production is catalyzed by the F1 subunit, whereas F0 is acting as a channel which is going to bring back the protons. So this is known as the electrochemical gradient, which is leading to the proton motive force, the force which is promoting the movement of protons across this membrane and helping us to produce ATP. Why is the proton motive force being produced in the first place? Because of the electrochemical gradient. So once the proton motive force is generated, now the cell knows that it has to bring back the protons, and how it occurs is with every movement of or with every proton coming back, the F0 F1 complex will move like a pump. So the F1 F0 complex will move slightly with every proton that is coming in. Once 4 protons come in, it produces enough energy to produce one ATP. So it works like a pump, it works like a motor or like a turbine. With every proton moving back in, the turbine is going to move a bit. With 4 protons coming in, the turbine has moved enough, or the F1 F0 complex has moved enough to produce ATP. So for every 4 protons coming back, I get one molecule of ATP. Now, if we see our energy yield, our net energy yield from glycolysis, from link reaction, and from the TCA cycle, for every 4 protons that came in, I got one ATP. But I had NADH2, we'll just go back to the previous diagram. I had NADH2 pumping out 10 protons, 4 here plus 4 here and 2 over here. That means a total of 10 protons are being pumped out for one molecule of NADH2. Now, when 4 protons of that comes in, I get 1 ATP. When 4 more come in, I get 2 ATPs. But the remaining are only 2 protons. I had pumped out 10, 8 have come back, I've got 2 ATPs. So the remaining 2 protons are considered as half an ATP. Of course, we all know that half ATP is not going to be produced. It is just for the mathematical calculation that we assume that for every NADH2 that is getting oxidized, I get 2.5 ATP. How did the 2.5 ATP come about? I had 4 protons coming back in giving me 1 ATP, 4 more coming back in giving me 2 ATPs, and the remaining 2 protons are half of the 4, so it is giving me an half ATP. In the same way, when we look at FADH2, FADH2 gives out only 6 protons at complex 3 and at complex 4. So 4 goes out over here, 2 goes out over here. Now, because only 6 protons are being pumped out by one pair of electrons donated by FADH2, because of that, the ATP yield from FADH2 is 1.5 ATP. How does that come? 4 protons, when they come back in, I get 1 ATP. 2 more are left out, so I assume it as half. So when we see this number, we can say that the net energy yield from glycolysis from link reaction and citric acid cycle is 32 ATP. How did I get this number? How did I arrive at this number? For every FADH2, I have FADH2 will give me only 1.5 ATP. So for every FADH2, I have multiplied it by 1.5 ATP. And every NADH2, I have multiplied it by 2.5 ATPs. So when you take that count, so along with that, I also get ATP from substrate level phosphorylation. So when I total all of it, I get around 32 ATP. Now, in the older books or in the older research papers, you will see that three protons are helping us to produce 1 ATP. But the current research, the most latest research is telling us that four protons have to move back in to get 1 ATP. That is why we have got this number. Some of you might have had your teachers telling that we get 36 ATP or 38 ATP, or you might have read it in books. That is also not incorrect. That is because it is taken with respect to the older number, where we assume that 1 NADH2 is giving me full 3 ATP. So based on that count, they might have told you as 36 or 38. But if you see the actual calculations, or as per the current research, it is only 32 ATP from one molecule of glucose. So every molecule of glucose, when it undergoes the aerobic respiration or complete breakdown, gives me a very good quantity, a very high amount of 32 ATP. This is not seen in any of the other pathways like anaerobic respiration or chemolithotrophy. We don't get this much of ATP. Maximum ATP yield is obtained when glucose undergoes complete breakdown by glycolysis, link reaction, and TCA cycle, and those reducing equivalents are further pumped into the electron transport chain and oxidative phosphorylation. I hope you are all clear with today's class. In case you have any doubts, you can always comment in and ask me the questions. I will definitely reply back. See you all in the next lecture. Thank you.